Okay, let's go ahead and get started. All right, so welcome to our first webinar for the ISPNE Early Career Scholars. There was an ask for this, so that's what we're providing is a couple of best practices and information for topics that are relevant to the society. So today we are going to focus on best practices for choosing biomarkers, research and collection considerations. Okay, so first we're going to go over some basic definitions. We're going to go through an overview of commonly used biospecimens for hormone research, including pros and cons. We're also going to look at time frame of what each biomarker captures, as well as biospecimens. We're also going to look at different assays used for extracting hormone data from biospecimens. That's going to be brief and towards the end. Quickly talk about data handling, especially since we're going to have a few of our other webinars cover this in depth. And then we're going to talk a lot throughout this entire thing, not just at the end, on how we decide what biomarkers and what biospecimens that we use in our experiments. I have a couple of really nifty little tables, so it can, break everything down in a quick table that you can look at, ranging from collection needs and costs to the times that are captured for each biospecimen or biomarker, and also ease of statistics. So ease of statistics can be rather relevant depending on your expertise. Okay, so first and foremost, what is a biomarker? Throughout this lecture, whenever you get these slides, there are tons of citations. That's one thing that was very important to me whenever putting this together. So don't feel the need to write down every little thing that you see. You will get the slides and you will get the citations that go along with this. But a biomarker is basically a measurable characteristic of the body that informs us of a biological process. So it can be a normal process, such as just what your blood pressure is. It can be a pathology, such as high blood pressure, high BMI, or a gene associated with a specific type of cancer or disease. A biomarker is not a feeling, it's not an emotion, it's not something that can tell us how a person is doing. It is a marker that is associated with a measurable biological process. Okay, so what is a hormonal biomarker? That's something that we're mostly going to focus in on this talk because we are the Society for Neuroendocrinology. So a biomarker, specifically a hormonal biomarker, is a molecule that informs a construct. And what I mean by that is it is a molecule that we think through prior research can tell us something about something else. So whether that be love, whether that be stress, whether that be how the HPA and HPG axis are playing together, it is a molecule first and foremost. Another reason that I say that is oftentimes just because we associate a molecule with a certain construct doesn't necessarily mean that the molecule feels the same way that we do about it. Take, for instance, oxytocin. There's a ton of research on the so-called love hormone out there, but I always remind people that oxytocin isn't just associated with those cuddly feelings. It's also associated with stressful situations. So keep in mind, first and foremost, that when we talk about a biomarker, we're talking about a molecule. Okay. Hormones are molecules that are released in the bloodstream to impact an organ, often at a distance from the organ that releases the hormone. The purpose of a hormone is often to trigger the release or the manufacture of typically another hormone. The action of hormones likely occur within cells, especially cell nuclei, where basically their function is to change gene expression across a wide variety of timescales. So we can be talking about minutes to years. A single molecule type can operate as a neurotransmitter if the conduit is in the blood. So think of that as in terms of like a pheromone or a targeted organ. So basically a pheromone isn't just something that can affect us as a person, but it can also target an organ in another person or another organism. Okay. Hormones are indiscriminate chemical messengers. And what I mean by that is they're not only received at sometimes like one specific receptor, and the message can be received by receptors across multiple organs. So they go everywhere and they do a lot, though very rarely do they just do one specific thing. So even if we're thinking about cortisol, and how that can affect both the brain and certain areas of the body. Sorry, the brain, neurological, the brain and other organs in the body. It's all the body. Hormones provide the substantive biological measurements that are capable of forming us of actions, habits, emotions, and characteristics deeply rooted in social and behavioral sciences. So I say that, but I also go back to my first point. Just remember that first and foremost, it's a molecule. Okay, so these are just a few examples. So one thing that I always like to note here is that when we're talking about hormones, keep in mind that, again, they are a ligand, right? So they are any type of molecule that produces a signal by binding to a receptor site on a protein. Hormone effects can be inhibited or regulated by competitive ligands. So again, going back to interplay between, like the developmental systems and our stress response systems. But overall, in general, they are broadly divided into peptides, steroids, and tyrosines. So hormones are needed usually in small amounts. They have very low molecular weights, and they are secreted at one site and typically act on a different site. They usually get used up in the process of whatever action that they're causing, and they regulate various actions and processes. So this is a point that I debated on saying, typically their actions are irreversible, but that's kind of true and not true. They're irreversible in a sense that, once they're released, but also other hormones can counteract or change the way other hormones behave. So these are just some typical hormonal biomarkers that we see a lot in the literature: testosterone, cortisol, and other glucocorticoids. Keep in mind that our main focus is going to be on humans. I will never forget as an undergrad, reading cortisol and cortisone and thinking that it was a completely different thing when I was looking at mouse models. But typically when we're talking, about humans, we use the term cortisol, and in mouse models, we'll, use a different type of glucocorticoid. And that's just typically because it's based off of which one is most abundant in what we're looking at. So we also have DHEA. These steroid hormones are smaller. One of the reasons that I like to mention this is because, since they are smaller, they do a really good job at crossing the blood-brain barrier. Then we have our peptides, which are generally a little bit larger. So oxytocin and vasopressin, leptin, insulin, also a hormone, GH, which a lot of people look into, and even melatonin is a hormone. Another thing that I want to mention is that this is kind of important when we're talking about deciding on what biomarker or what biospecimen that you want to use in your study. So biomarkers, aren't always, they're not always hormones, one. And as I said earlier, they're not always necessarily molecules. So of course we have things like cholesterol, telomeres, markers of inflammation, genes, things of that sort. But also keep in mind that biomarkers can be things such as a person's blood pressure. It can be their BMI. When we think biomarkers, we don't want to necessarily just limit ourselves to thinking about a molecule. And again, that's just really important when we're thinking about the questions that we want to ask. So for instance, if we are interested in studying stress, think about other things that can be affected by stress when you're picking your biomarker. Things like blood pressure, things like cholesterol, things like weight gain, all of these things can relate back to stress, but it's not necessarily something that we measure in a biospecimen. So let's just go, because I've used this term biospecimen a lot. Biospecimen is very simple. It's a quantity or a tissue, quantity of a tissue, blood, urine, or other biologically derived material. Basically, it comes from the human body. I put this here because it can be anything ranging from skin to plasma to blood to urine to hair and to nails. And again, for this lecture, we're mostly going to be focusing on the pros and cons of human biomarkers and biospecimens. But again, a lot of these issues can also occur in non-human models. Okay, so the first biospecimen that we are going to focus on today is blood, plasma, serum, and serum draws. So blood, as well as serum or plasma derived from blood samples, is a very well-established biospecimen for assessment of human hormones in research and medical settings. We've been using it for quite some time. Not a lot of things that are novel about it. It's a solid biospecimen if you're looking to look at certain biomarkers. Also, depending on the hormone or the molecular structure, so especially when we talk about our larger hormones such as peptides, blood may be your only option. We'll talk about oxytocin specifically a little bit more later, and we also have a lot of citations for this, but to this day, there is still controversy on whether or not we can get larger peptides from saliva. So to be safe, depending on what your research question is, what you're focused in on, it might be a good idea to use a blood draw. This is also quite an invasive collection. I mean, obviously it's not going to be as invasive as like CSF, but it is something that can cause some discomfort in our participants, especially when we're talking about human participants. So of course, there are a ton of blood draw pros. One, it captures many hormones ranging from peptides to steroids, and there's not controversy surrounding that. We know that we can get some of these larger hormones when we talk about blood and blood draws. But you can also get some of your smaller hormones such as testosterone and cortisol. So again, I'm going to repeat this. It may be your only option depending on the hormone that you're looking at. Again, it's a lot of controversy on whether or not these really large hormones make it into saliva. The other great thing about blood draws is that you can capture other biomarkers. So you can look at telomeres, you can look at cholesterol, you can look at total blood count, and you can also look at genes. You can look at acute as well as short-term changes in rhythmic qualities in hormones when using blood draws. And it's also stable for a fairly long time, depending on the marker that you're looking at. So when we're talking about, if you're going to look at hormones, but also just look at red cell blood count, these are stable for up to 10 years, if not maybe a little bit more, and negative 65 degrees. Another great thing about blood draws is that large samples can be easily aliquoted for disbursement. So aliquot basically means you have this, this nice sample of blood or multiple draws of blood. If you go on to decide that you want to look at something else, or if you're a lab, if one lab's going to look at steroidal hormones and you want to send it off to another lab to look at cholesterol or total blood count or whichever else, it's very easy to aliquot samples and send them off. Not necessarily something that's easily done with saliva. And, in saliva, you're not going to be able to get some of these things like total blood count. So there are a lot of cons as well. Unfortunately, you're going to need probably a phlebotomist to do the blood draw. You have to worry about fear of needles. So that can affect the willingness for your participants to actually participate in these studies. Parents might not be as willing to allow for blood draws in their children. And kids may not want to, they might not want to do it repeatedly. So even if we, I've been involved in studies where we've had no problems with getting that first sample, but then they come back, especially if the time point that you're looking to do the collection is, within days as opposed to months, they don't always want to do it. Also, you have to consider temperature controlled storage and transfer. So it's not something that you can just, put into a box to wait to get back to the lab to freeze it. A lot of times, depending on the biomarker that you're looking at, you do have to have your blood in a temperature controlled, storage, temperature controlled storage for transfer. It also does carry, depending on where you are and how much the sample is, a biohazard tag. Oops. So again, with blood draws, it requires usually phlebotomy training. I say usually because if you're working with a doctor, you don't are already, then you don't necessarily have to go through the training or find a nurse or someone who's done it. It does require specialized equipment. So getting the butterfly draws or syringes, things of that sort. And depending on what you want from the blood, degradation can occur in about 24 hours or so. Okay, so that was invasive. I do want to say that there are certain biospecimens that we're not covering. So like cerebral spinal fluid, that's not something that we're going to cover because typically we don't use that in behavioral research because it is such an invasive process. But that is another biomarker or another biospecimen that can be used to look at hormonal biomarkers. All right, so next we're going to talk about two minimally invasive ways of getting some of these biomarkers, and that's through dried blood spots, as well as an emerging technology called TAZO. Oop, I think this got mixed up. I'm sorry about that. This was supposed to be in the last slide, but this was one thing that we do have to worry about with blood storage. Oh, sorry, this isn't in the wrong spot because we have to consider this for DBS as well. When we're talking about blood draws as well as DBS and a few of these other things, ultra freezers are definitely needed for long-term storage and refrigeration for short-term storage and transfer. Let's see, with blood spots particularly, they don't have to immediately be refrigerated because you want them to dry, which we'll talk about a little bit later. Okay, so dried blood spots. I'm just going to refer to it as DBS from here on out. But DBS is a sampling technique that involves collecting a very small amount of blood from a study subject. The blood is typically spotted on cellulose or polymer paper cards. You can see them, they can come in like little 4-packs. So I don't know if anyone's ever seen a baby get tested for jaundice. So typically the way that they'll test that is through a dried blood spot where they collect it and put it on a piece of paper. And the specimens are then dried at room temperature for 4 hours before being shipped to a laboratory for analysis. And the ideal situation, if you're reading the literature, you would put it underneath the hood and let it dry for 24 hours. But typically, approximately 4 hours is considered acceptable for letting them dry before being shipped off. So some of the advantages are that DBS is, again, right up there with blood as far as being very well established. So it's been used for over a century, and it originated as a method of observing blood onto paper to measure glucose levels. It's very cost effective, so you're eliminating the need for a phlebotomy personnel because you're not doing venous blood draws, and also equipment costs. So it can be completed in a wide variety of settings, which is wonderful. Not having to do a full vein puncture means that you can use it in remote contexts where freezing or storing samples on ice is impractical, especially if it needs to be done immediately. Typically, once these are dried and sent off, you're good to go. But for long-term storage, you do have other things to consider as well. So room temperature storage and shipping has made DBS the go-to specimen for biological anthropological work and field work. And you get a wide set of potential biomarkers as concentrations are somewhat comparable to blood sampling, but keep in mind it's really small. I actually just read a paper where they were using DBS from babies to look at telomere lengths. But one thing that you have to consider, and one of the critiques that you get is there going to be degradation in that sample if you're just using a dry blood spot? How quickly was it dry? Did they let it dry for the full 24 hours? How long has it been in storage? How was it stored, et cetera, et cetera, et cetera? So these are some things that you do have to consider when you talk about DBS as opposed to a full-on blood draw. So some of the cons is, granted, it's not a full-on, blood withdrawal, but that prick can be pretty painful. I don't know if any of y'all have had the thumb prick, but it's painful, and also the anticipation of the pain can be exaggerated based on the perception of how invasive it can be. That little puncture, it hurts. It especially hurts whenever you have to do it multiple times. So, thinking of whether or not a person, if you have to collect a lot of blood, is it easier for that person just to get one big draw done where you can have a nice large sample of blood? Or depending on how many pricks, how uncomfortable are they going to be? Because that can be really sore to do that multiple times on a thumb or finger as opposed to just doing a single draw. Again, that telomere example, some biomarkers are degraded on the filter paper. So you have to be very cognizant. You have to think about what biomarkers that you want. So as opposed to blood, if you later decide that you want to adjust your IRB to look at, X, Y, or Z, you have that vial, you can get it. If you later decide that there's a control or there's something else that you might want to get from the DBS biospecimen, then you might not be able to get that. Extracting blood from the filter paper can be very time consuming in the lab. So again, if you are wanting to look at a bunch of different types of biomarkers, it again might be a good idea to get that larger blood draw. Insufficient saturation is a very common source of error. So that's something that you definitely want to look at, making sure that it's saturated into the paper and also making sure that it's properly dried. So you think about if you're working off site, but you're working in a very humid environment, is it going to actually saturate that paper, and is it going to get dried enough so that you're not going to have that error, especially if you don't have access to a vent hood where you can let it dry for a long period of time. Okay. You can actually get DBS collection from a venipuncture from the vein. So if you're taking a larger sample, you can also send off some of the a DBS just by aliquoting, I guess, for lack of a better word, from that larger sample. But typically we see it in skin punctures. So whether that be the finger, arm, or the heel, it's typically a skin prick. And spots usually have to be prepared in less than 24 hours. And again, we already mentioned dried overnight or at least for 4 hours. Okay, as far as storage is concerned, for that initial transfer, typically they're stored in a zipper bag with desiccant satchels. And the reason that you do that is because once it's dried, you don't want moisture to get in. You can also include a humidity indicator card. That's actually a really good idea for using, to eliminate errors to figure out if humidity got to your sample. So do both. Okay. So again, for that initial transfer, It's fine, that you're just using the desiccant, it's dried, you have the humidity card, but as soon as possible, you do want to get it at least negative 20 degrees. So when freezers are not available, so under field conditions, storage at negative 4 degrees Celsius or even an ambient temperature is feasible for up to 14 days. But again, depending on the biomarker that you're looking at, that might not be the best idea. So transport, one of the best things to do is just to transport it frozen, especially on dry ice, again, when we're considering things like humidity in the equation. So no biohazard marks are needed. I was actually very surprised by this within the US whenever you're doing DBS, but the biohazard mark is needed whenever it's going through international shipping. So going back to point one, those desiccant packs and that humidity indicator are really important because if there is evidence that humidity has reached your sample during transport, it's a good idea to exclude them. Okay, so the next minimally invasive thing that we're going to talk about is TAZO, which I like to just call a fancier version of DBS that gets you a bit more blood. So TAZO is a proprietary, sorry, I can't speak, method that is usually attached to the upper arm or hip. And it's really kind of cool because it avoids the sensitive nerve endings that are located in your finger from doing those pricks. So as such, it's typically reported as being less painful and stressful. TAZO collects basically the capillary blood over a small span of time, and it allows for smaller punctures for equivalent volumes compared to DBS. They also are very convenient. I mean, they send you pretty much out the whole shipping package. More importantly, participants can easily collect it on their own. Not to say that they, couldn't do a finger prick, but just thinking of, whether or not you can trust them to dry the finger prick for 4 hours, that it's not gonna, get set on a counter and, someone spills juice or something on it. It is something that they can follow the instructions for. And since it's contained in a little device, you don't have to worry about that contamination. Also eliminates the risk for bloodborne pathogens since they're not exposed to the blood as they would be with TBS. And the lances aren't visible. So for people who have needle phobias, especially like pediatrics, but I will note, we'll talk about this a little bit later, it hasn't been fully validated in pediatric work. And also vulnerable populations, since they don't see the needle, it's possible that they're not as nervous or afraid about, performing the blood draw. And of course, one of the advantages is you can collect quite a bit more than you would with a regular DBS. So some of the cons, it's still considered novel and it's still considered an emerging collection method. And there are a limited number of validated hormones. Capillary blood samples take minutes, not seconds to collect. So there's an inherently long time window of the hormonal metrics. So depending on the time point that you're looking at, it might not be the best choice because it's a slow collection as opposed to that quick thumbprick and getting it done and getting it dried. The volume of blood collected is necessarily small, so large panels of hormonal biomarkers may be impractical. I mean, just think about, go look at your assay sheets. How much are you going to need to assay, 4 hormones? Are you going to get enough based off of that 200 UL? And are you going to be looking at other biomarkers as well? So if you're looking at cholesterol, you want to look at total blood count, you want to look at all these other things, it might not be practical to use this technology. Okay, So short samples may be especially prevalent in pediatric populations or other populations where a person has a low blood flow. So, as opposed to with a thumb where you can just pinch it and get that, blood flowing, get it on your DBS, you might not be able to get, you might have some error, you might not get as much blood as you think that you would using this, and there's really no way that you can tell until you get it back in the lab. And it's yet to be validated for use in pediatrics. Again, think about populations with low blood flow. So when multiple samples are needed within one experimental setting, you need multiple TAZO kits. So this adds expense and also multiple punctures, which your participants might not like. Again, it could have a negative effect rate on research consent rate, especially if you're thinking about a longitudinal study where you have to take multiple punctures or the participant has to do multiple punctures. Is it easier and is it financially, a better idea just to have the participant come in and do that one blood draw that gets you everything that you need? It depends on your question. Also, shipping and storage. So how far are they going to be shipping it? Is it going to be cold enough depending on the biomarker? That can be a little bit tricky. Overall, in general, the way that the TAZO collection works is it sticks to the skin with this little, it's adhesive, so almost like sticky tape. They push the button, a vacuum forms, the lancet comes out and pricks the surface of the skin, draws it in, they ship it away. So this is, it's stored in the device and not climate controlled. You can put an ice pack or something in there with it. But overall and in general, it needs to be shipped rather immediately, depending on your marker of interest. As far as working with people and humans as a population, we all know that sometimes the words ship immediately are not followed. So that's something that you absolutely must consider if you're considering using the TAZO method, as opposed to a blood spot, which is dried, or just having them come in and getting a full blood, doing a full blood draw. Okay, just keeping an eye on our time here. Let's see, before we move on, because I haven't checked the chat at all, before we move on to non-invasive, are there any questions whatsoever about minimally invasive and invasive forms of biospecimens, biomarkers? No? Okay, and if so, if you do have any questions, just go ahead and put them in the chat.
I have a question, Dr. Moody.
Sure.
I was wondering if there was a difference between plasma and blood versus serum and blood, and if there are different biomarkers found in, those things.
Yes, especially so depending on your protocol, I typically will use salometrics in the labs that I've worked in. Depending on your protocol, you might want to use one or the other. And so like, let's say you're looking at oxytocin, and you have to go through the extraction process. It might be a little bit easier to do that extraction process just using plasma than with blood. It just really depends on what you have. And if you're looking at different biomarkers, so if you want to look at cholesterol versus total blood count versus oxytocin versus steroid hormones, and you're aliquoting them separately, that's going to make a difference on who gets what. But generally, you send the whole blood sample, at least that. Well, I've seen it where you can either just send the whole blood sample, depending on which lab is looking at which biomarker, or you can also separate them and just send plasma. But you're going to have different protocols to follow depending on the biomarker. So you can divide it and send it out, or you can just send the whole blood sample. It really depends on how much you have. But yes, you're going to get different biomarkers. You can get, the same biomarker from plasma versus blood, but depending on your protocol, what assay you're using, what technology you're using, that's going to make a big difference. Does that make sense? Did that answer your question?
Yes, thank you.
Yeah, so again, just to reiterate, you can break it down and ship it out. And, in case you have separate labs looking at different things, and you are going to get different biomarkers from, whole blood counts to cholesterol to steroid hormones to oxytocin, depending on if you're using the whole blood sample or just the plasma. Don't correct me if I'm wrong, but most, or just don't correct me. Correct me if I'm wrong, but like oxytocin protocols, they'll have protocols for like plasma or for whole blood samples, if you don't want to break it down or if you just don't have enough sample to break it down. And the extraction protocols definitely differ depending on what part of the blood you're using. So just because I don't have them all memorized, make sure that you're looking at your kits before you do those draws. That's one thing that I highly recommend if you're jumping into hormone research and biomarker research. All of the kits, unless you're making your own kits in your lab, or making your own assays in your lab, most of the kits that you use, the commercial kits at the beginning, will have how much plasma you need, how much blood you need, how much saliva you need, depending on which one that you're using. Whether you need to do an extraction process on one of those biomarkers. So it's an important consideration to read those protocols if you're doing them in-house. And if you're not doing them in-house, just call whatever lab you're planning on using, and ask them how much sample are they going to need. Not just, they should tell you how much they need for an initial testing, and also if they have to perform like we call them redos in our labs, but if something goes wrong with it, how much sample do they need to run that again. Typically, if I'm thinking of an experiment in the design, I'm not just thinking of one sample, I'm thinking of something's going to go wrong maybe in the lab. Is there enough material for them to run it again? So look at your protocols, talk to your lab, and see what they need. But that's a great question. And we'll talk about it a little bit later, but also think about the design, think a lot about the design. So if you're doing diurnal, reactive, what have you, but we'll get into that later. Okay, so now we're gonna get into non-invasive. If my screen would like to advance, that would be great. Okay, so the first non-invasive biomarker that we're going to talk about, which a lot of people are probably familiar with, especially in developmental research, is saliva. So saliva is often collected through passive drool, and that involves directly expectoring into a vial or tube. Basically, in short, it involves directly spitting into the tube. Alternatively, saliva can be collected with the aid of devices such as straws, larger collection funnels, or absorbent cotton and polyester, and other types of filters. I will say immediately, before we continue, be very, very, very careful whenever you're using cotton, polyesters, and other types of filters, because it can cause some trouble depending on the biomarker that you are looking at for your lab. It's always best, unless your population isn't able to do it. So think about children, young children especially, that we've tried to think about a few ways to get spit from babies, like whether it be, those suction machines that they use at the dentist, collecting the saliva that way. Unless they are unable of spitting into a tube or a vial, that's the only way that I would consider using absorbent polyesters and filters. Just get the saliva whole if you can. So some of the pros is saliva is non-invasive, and it allows for repeated samples that can take place in a variety of settings. So again, thinking about diurnal, like if we're looking at diurnal processes, or if you're looking at multiple collection points throughout the day or an experiment, it's a lot easier to have someone spit into a tube multiple times or agree to spit into a tube multiple times than to do multiple blood draws throughout an entire day. Ease of collection also means that you can have participants do it by themselves. So that includes the home. Depending on what you're looking at, though, this is, it's easy, but you do have to worry about things like time. So when they take the sample, and we're going to go over that more when we talk about diurnal rhythms, but when they take the sample is really important depending on your developmental, or that shows my bias, depending on your experimental question. So again, timing of the collection is crucial. Dry mouth, which can be increased when people are stressed. So I've seen it happen myself. If people are just, kind of anxious and you're taking a long time, people can become a little stressed out. And so that increases dry mouth, and that means that they have a lot of trouble spitting into the tube, or sometimes you'll get like a lot of bubbles. So you're having to tap it down a lot, and it can take them a long time to fill up the vial, which of course makes them that much more stressed out. Multiple samples can lead to missing data. So, on one hand, it's very nice whenever you can have someone go back home and do the experiment and take the samples at a certain time point. But if they miss one, or if the timing's too off, it can definitely lead to missing data more often than I would like to see. One thing we also have to worry about, which will absolutely change what your results are, is contamination. So things such as blood, so people who might have certain medical conditions, or if a person, forgets and brushes their teeth, or eats and, scrapes their mouth or something on a chip, and they give you that sample, you can get contaminations of blood, which is going to cause a completely different outcome whenever it's ran through the lab. And so your samples can get contaminated in that way. If you're in lab, embarrassment. So, I've seen lots of people giggle, but, there sometimes can be a little bit of embarrassment because there can be an odor, the saliva spilling over the side of the tube, things of that sort. So it can be very expensive to ship depending on the biomarker that you're looking at. So, they do have to be frozen. If you're going to wait to ship all of the saliva samples at once, they have to be kept frozen, preferably in a negative 80 degree freezer, but, definitely in the negative 20. And it gets expensive because dry ice is heavy, making sure that it's properly packed, making sure that you're sending it overnight. So you just can't send it by regular mail. And also expensive in terms of storing it on site, especially if you don't have access to like a deep freezer. Ask around though, especially if you're in a university setting, someone will usually have a deep freezer that you can use to keep them cold. Also, you're limited in your biomarkers. So it's great for steroidal hormones, definitely some inflammatory markers that you can get from it, but you can't get cholesterol from saliva. So again, passive jewel is the best. People oftentimes will use straws or wider mouths to collect in the tube. So you'll see different companies will have like a wide, almost funnel type thing that can go into the vial. And then that makes it a lot easier for people to spit as opposed to them, aiming to get it in a very tiny tube. Depending on the biomarker, again, avoid non-passive jewel methods, so cotton balls that could cause contamination. And this is one point I should have just made a slide that was just this. As a matter of fact, I think I did. Cheek swabs are not passive jewel. Cheek swabs are great for genetic material. Absolutely wonderful. It is not passive jewel. And I say this because a lot of times, so not only cheek swabs, but, or when people are doing the cheek swab and then getting like a little bit of saliva in the little cotton scrapers, those go into saline solution to keep the sample preserved. And you are not going to be able to get reliable results from that for a steroidal hormone marker. You're just not. The sample pretty much is contaminated. You're going to get genes and genetic information all day. It's great. But I do want to stress this because I've unfortunately seen it happen a lot where someone has been very excited and they're like, I'm going to get, testosterone and cortisol, and we did these cheek swabs. It is not not recommended that you use those. You're just not going to have enough saliva. You're not going to have really any saliva. So do not make that mistake. So for transfer, typically they're in these little tubes, and if you do have a tube like this, tape it down. But typically you'll have the screw-on tops for transfer, making sure that they're labeled and things of the sort. Depending on the biomarker, you have to refrigerate it almost immediately. I know Emma spoke a little bit about this in her talk, but there are companies out there who, if you're looking at cortisol, they don't have it refrigerated at all. There are commercial companies that say, put it in a packet, ship it across, and we will get it. I do caution against this though. Even though cortisol is an extremely, it's a very hardy hormone, if you're also going to be looking at testosterone, estradiol, DHEA, you must be very, very, very cautious with not having a saliva sample, sent after being frozen or on ice. Typically, again, on dry ice. So yeah, you might get cortisol, but then maybe later you decide to look at testosterone and how stable is your saliva sample if you're not sending it frozen. So again, keep in mind that frozen samples do have to be sent on dry ice. If it seems like I'm harping on this a lot, there is some literature and there'll be some references in the notes section when you get the slides. There are changes there are changes in the concentration of certain hormones, especially testosterone and estradiol, depending on the freeze-thaw cycle. So when I managed a lab, one of the things we would do is make sure you get those, if you know that there is going to be a freeze-refreeze cycle, make sure you get those less stable hormones assayed first. And so you just don't want that to happen. You don't want to have it thawing. You don't want to have it overheating because, I don't know, maybe it was just a really hot day. Delivery trucks are not necessarily insulated. So you do want to send them frozen if you can. Again, I talked about that already. Freeze-thaw cycles are definitely something that you want to consider, whether that's in lab or whether it's during shipping. You also have to consider long-term storage. Temporarily putting it in that -20-degree freezer is all fine and dandy. But if it's at -80 degrees, it's going to take longer for it to unthaw. So if you're taking samples out, organizing them, it's going to take a lot longer for it to unthaw at -80 degrees than it is at 20. And so they'll give you a little bit more time. You definitely do not want to do long-term storage in, a regular freezer. You definitely want to get it into those -80 freezers. And also, I just put this in here. This is and I've seen it happen with so many different experiments. You have blood, and typically whenever you're doing an experimental design with blood, you have 1 or 2 vials. If you're looking at saliva, and you're looking at diurnal rhythms, it is so important that you keep those very well organized. One of the advantages of saliva is that you can get those multiple samples. But if you're not organized and you have sample 2 marked as sample 1, that's going to cause a lot of confusion on a lab end, and it's going to cause a lot of confusion on your end. So again, while it's all fun and dandy that we can get 4, 5 samples in a day, 12, 24 across 2 or 3 days, you want to make sure you keep things organized. You can't look at one saliva sample and another and tell what time they took it. Okay. Does anyone, because I know that was a lot, does anyone have any questions about saliva before we move on to urine? Okay. And again, if so, just place them in the chat. So urine is great. I mean, it's a minimally invasive way to capture hormones over a cumulative period of time. So keep in mind, it's not like a just-in-time hormone. You're not going to be able to necessarily do an experiment, and then get immediately what the hormone does with urine. You don't know when a participant has to go to the bathroom. So it's typically good to think about, think over like 24-hour periods of time or for a set number of days. So urine is ideal for research seeking to examine larger molecules as well as steroidal hormones. So, kind of going back to oxytocin and saliva and some of the controversy there, is really, urine is a great way to get at peptide hormones. So if you're interested in a peptide, especially over a longer period of time, so more of a, a trait or a day-to-day state type of measurement, then you definitely want to consider urine as a non-invasive means to look at that. Also, urine is a relatively copious amount. That's a fancy way of saying you get a lot of it. So if you want to look at a lot of different hormones or do, these huge panels of hormones, urine is the way to go. So there are some pros with urine, again, minimally invasive. Most people have provided a urine sample before, so they're comfortable with doing it. Again, large amounts and large molecules. Cumulative, so depending on, again, the biomarker, your experimental design, you get just a great marker over 24 hours. Some of the cons though, so it can be very difficult to collect in certain populations. So, thinking of children, I've seen some protocols with diapers, but still, it can become contaminated with feces, things of that sort. So definitely difficult whenever you're thinking about your population, even animal populations. It does have to get stored pretty immediately in cold temperatures, and that's to avoid sample degradation due to microbial growth. Again, keep in mind that this is going to change depending on what you're looking at. If you're looking at growth hormones, if you're looking at oxytocin, if you're looking at steroidal hormones, sometimes that time, there's a little bit of wiggle room there, but you generally need to get it stored immediately in very cold temperatures. Also, you have to worry about blood contamination with urine as well. A little bit of blood can cause a lot of change because typically what we see, hormones that we see in blood are going to be typically far more concentrated in blood than we are going to see it in urine and saliva. One issue is that fluctuations in hormones will happen if large amounts of water are consumed, and that's kind of relative, right? What one person might consider a lot of water, another person might be dehydrated, which can also change your concentrations. So that can add a little or a lot of variability. And depending on your hormone, however, there are ways to get past that. But that can get costly. So, if you're looking at oxytocin, you might also want to look at vasopressin and, oh goodness, okay, I will put this in the notes later, but there's another hormone that you typically want to look at that can tell you how much water is in the system. So that way you can use it as a control variable whenever you're looking at these larger peptides. So that can cause a little bit of cost and, depending on your reviewer, they might want to see that you use that as a control. There is a little bit less reliability due to the hormones being metabolized prior to urination. So, especially considering your time scale, you might want to go, especially depending on the experiment, with saliva or blood or something else, because you're not, again, going to be able to get it immediately. And typically it should take place in a lab setting. Again, this goes back to, trusting your participants. Are they going to refrigerate it? Are they going to send it right away? It has a little bit less wiggle room whenever you're considering how cold it needs to get and how quickly. So urine collection, pretty straightforward. Most of us have probably done a urine sample. You need a cup with a top that can be secured, usually a bag to put that in if you want double containment. Make sure it's immediately cooled, and samples are best taken midstream. We say that because usually that first stream, I don't know how many of you have taken a urine sample, you usually get rid of just like a lot of interference or a lot of air that can be caused for bacteria on the outside of the genitals. So you use the restroom a little bit, take the sample, and then pack it up. So you do have to worry about folks following those instructions as well. And again, it can be more or less important depending on the biomarker that you're looking at. Okay, that's pretty much saying the same thing. So we're going to pass that. The next non-invasive hormone that we're going to look at is hair. So hair has grown into an established biospecimen that can be collected non-intrusively to measure cumulative hormone concentrations. And this is approximately at a rate of 1 month's worth of time per centimeter. There's a lot of approximation there, especially when we're thinking about the fact that hair can grow at different rates depending on the season. Depending on the health of the person, depending on how much stress they're under. And there's a lot of validation work that's still going on with that. So we say that it's approximately a month's worth of hormone in a centimeter. So the good thing about hair is it is relatively simple and can be contained in a lot of different settings. I have had people collect it at home. You can collect it in the field. You can have them come in and do it in the laboratory. It's pretty straightforward, and a lot of the equipment that you need to collect it, some people have at home. So usually a pair of scissors, foil, making sure it's labeled, a rubber band. You don't have to get like the specialized urine cups, with making sure that it's firmly sealed. So it remains stable over long periods of time at room temperature and not at room temperature. So you can safely ship it. And we've had some questions come up, and there's a couple of studies out there that look at, temperature changes. So think about if you're having a participant send a sample during the winter and we have a freeze and your samples get caught in transit, which may or may not have happened to me a few times, or especially if you're, transferring it international. It does tend to stay stable, and there's not a lot of degradation in hormones. It's remained, hormones have remained detectable in hair over millennia. So that points to it being pretty stable. Now, I wouldn't want to do a comparative study where I looked at it, I don't know, one mummy versus another mummy that's in a completely different region of the world. But if everyone's pretty much been in the same conditions, hair has been another go-to for field research and research that deals with postpartum or historic samples. I think, was it most recently, I think they did use hair to look at, genes or something or another in Beethoven. So hair, they got it from his brush. So again, hair is pretty stable. I say that though, but keep in mind, whenever we're looking at steroidal hormones, it's, we don't necessarily want that follicle. So that has happened before where we get samples of hair back and they just pull it out of their brush. And so we might have a little bit of a follicle, and we don't want that. Typically just want as close as you can get to the scalp. It does eliminate a lot of financial fees and hazard costs from shipping with dry ice. So you just wrap it up in foil, make sure it's in a secondary containment package, and ship it away. In-home collection is feasible because it's easy to obtain hair collection tools. This actually this was extremely important during COVID for a lot of different studies that I was involved in, was that they didn't have to come in and we could pretty much talk them through it, whether it be on the phone or via Zoom on how to obtain a hair collection sample. It's robust against attrition rate, as ample information is gained with a single collection. So as opposed to, getting a state measure of a hormone over a month where you have to get multiple samples, one collection will get the job done. I say robust attrition rate, but as we get into pros, cons, we'll talk about something else there a little later. One good thing is that it controls for variations in hormone release, such as menstrual cycles, and sorry about that, such as menstrual cycles. So when we talk about diurnal hormones, we'll continue on this. But one of the things oftentimes that you'll have to control for whenever you're doing reactive hormones or whenever you're doing diurnal hormones is making sure that you have a basal day or a base day to compare it to. And you don't need that with hair. With that being said, it, as an advantage and a disadvantage, it captures chronic exposure to stressors or different hormones, or so it doesn't require multiple collections, but you can't get that snapshot. And it's also a con in a way, because if you're, thinking of your experimental design and you want to, X happen and then, Y happen, depending on what happened in that person's life in that month, typically 3 months is what we look at. You might not necessarily know what's causing that change because things happen in their life. So is it X that caused that change? Is it your intervention? Or is it because, there was some historical event that happened? And this is something that's come up in a few designs that we've had. So again, it cannot capture short-term psychological stressors or physiological responses. So, thinking of urine, we're thinking of, about a 24-hour or so period or even hours. When we're talking about hair, we're talking about months. Insufficient sample weights can indicate poor sample quality, and it will definitely result in systemic error. So, as opposed to, urine or blood or something of the sort, you usually have enough of the sample. It is, especially when people are self-collecting or you have researchers who are not familiar with the collection process or get nervous or skittish about, cutting a lot of hair. If you don't have enough, it's going to throw off your results. And so typically what we say is we don't want to look at 1 centimeter of hair. We want to look at 3 centimeters of hair. So instead of saying a month, I should probably say that you should be thinking in terms of months whenever looking at hair. So distal segments do not necessarily correlate strongly with proximal scalp region segments. And all this is to say is that if you're not having a person getting that sample close to the scalp, we know that once you get past 4 centimeters, there isn't a large correlation between the first 3 centimeters. Even though the term is not true, I usually tell participants to think of it as that first 3 centimeters still being alive and, 4 centimeters out, that's kind of dead hair. So, people will say, oh, I have a lot of hair, I have long hair. That's great. We're not using the entire length, you're only using that first 3 centimeters. Also, bias can be introduced from external variables. So that includes exposure to heat, chemical treatments, dyes, perms, relaxers, natural sunlight, and UVs. So this is one thing that I tell people, absolutely collect this data as control variables. Unlike with saliva and cortisol, salivary cortisol, salivary testosterone, and things of that sort, also blood, you want to make sure you're controlling for all of these different things. And also I say that, you can test these in your models to make sure that there are, they don't have an effect and then take them out later. But also it is a con that if you do end up having to control for these, think about your degrees of freedom and how that might change your experimental design. Also, there is evidence that hair type may impact results. So if you're, looking at groups where you're going to have multiple hair types, there could be a little bit of error introduced from that. Basically, saliva is generally consistently all the same. Hair is not, and that shape can affect how much hormone may be being absorbed into the hair type. Typically, we say that you should collect it from the parietal region of the scalp, less sun exposure, close to the scalp. You cannot use hair from a brush in the first 3 centimeters. What advantage, again, is that it is easy to transfer. So breathable container, generally room temperature, keep it away from light. And it can be safely shipped in the mail with minimal packing concerns. So if you're getting samples in from another country or, crossing state lines, it is very, very, inexpensive to ship the hair. That I did. I'm very happy that I put this in here. Again, one of the things to remember is that if you're looking at hormones, just don't do a cheek swab. For genetics, it's great. And we decided not to keep this in because if you have cheek swabs or if you want to do a cheek swab as a part of your design, do a cheek swab and also do a saliva collection. Okay, so this is some emerging, these are some emerging biospecimens to look at different biomarkers. Nails, fecal matter, sweat, and teeth, we took that out just because, again, it is very novel. So there's research on it, but it's not a lot. So it is something that you can use. A lot of the designs that are out there, and there should be a few in the notes, usually involve baby teeth. But it's not exactly the most, unless you're a baby and you're losing teeth, the most non-invasive tool to use when looking at biomarkers. So there are definitely other things to use, but teeth are one of them, but we're going to consider these three in depth. Okay, so some of the emerging biospecimens to use, nails. So they follow the same rationale as hair, and it is minimally invasive. So you can, again, capture a hormone level over a long period of time without much trouble. It's been successfully, nails have been successfully extracted for testosterone, cortisol, and DHEA. And they're used in a variety of assays, but typically, most commonly, we see them used with mass spectrometry. I am going to go back and look at this recording and laugh at myself for the way I pronounce that. So it is an emerging way to look at hormones. So some of the pros is it's, again, non-invasive. You can use it in post-mortem studies. You can also use hair that way, but I've commonly, when you see nails, I've seen it in that regard. When hair collection is not possible or participants might be unwilling, there are definitely times where I wish we were collecting nails as well, especially to reduce attrition. A lot of times, depending on the population that you're using, for instance, maybe teenagers, they don't like providing a large hair sample, especially if you're going to be looking at multiple biomarkers. So some of the cons is that, again, it's novel and it's limited in the, invalidated hormones. So they're, as far as like if you're looking at progesterone, I'm pretty sure that it hasn't been validated with progesterone. Or if so, there are very limited studies that look at it. So there are very few studies, and by very few, I mean one, that looks at external influences. So, things such as nail polish remover and how that might impact the hormone. And susceptibility to different growth rates, but it's approximately about the same as hair. However, with that being said, one of the things that we know about hair is that, again, we want to get the hair that's closest to the scalp of the participant. We don't want to go too far out. With fingernails, obviously, we cannot take a person's entire fingernail, away from the nail bed because you have, this much room, or about, I don't know, a few centimeters, probably more depending on the nail, more than 3. And so we don't really know how well. There haven't been a lot of studies that tell us how much correlation is going on between here, here, and here. That probably needs to be done in an animal study because I don't think people are going to let you rip their nail bed out. So that's one of the biggest problems that we have with nails. So again, when the hormonal deposits occur, how much degradation occurs, how well it correlates, from 1 centimeter from the nail bed versus 2, 3, or 4, it's very unclear. Also lifespan differences in growth. But again, you can also see this with hair. So think about differences in growth and hair growth during pregnancy versus non-pregnancy, as well as, nail growth during pregnancy. So you might see different differences in growth rate depending on the population that you're looking at and what life stage they're in. But nail collection is really straightforward. You click the same length, and you're good to go. It can be, kept in a Ziploc bag. There's not a lot of confusion or a lot of equipment needed to collect nails. Over the long term, cool, dry, breathable containers. It's also very easy to transport, so similar to hair. So another one of my favorite non-invasive forms of looking at hormones is feces. And it's a sample that is useful for assessing hormones in a complementary biospecimen that's basically, close to the microbiome. So if, depending on where your interests are, you can get a ton of different data when looking at feces. The total fraction of hormone represented in fecal samples may be included as a byproduct of the microbiome community. And although the range of hormones extends beyond cortisol, one of the primary advantages of fecal cortisol is it sheds a light on stress endocrinology and the microbiome. So again, when you're looking, you can look at everything, unlike saliva or even urine, there are some great gene sequences that you can do and different biomarkers that you can look at with feces that you can't look at with other biospecimens. And it also is basically in conjunction with biological stress. So experimental designs can connect glucocorticoids and other significant hormones to microbial analysis, and can also give us a little bit of information on internal microbial environment. So basically, again, the microbiome. It's potentially passive. So again, when you're thinking of, babies and, going back to how difficult it might be to get just a saliva sample that isn't going to get any air because you're using cotton swabs, or they don't really generally sometimes have a lot of saliva. It's difficult to get to, that it's passive. So, you can get it from a diaper. Samples as small as 1 gram, so a teaspoon, can be used to assess biologically relevant hormones. So you don't need a lot. But there are some cons, right? It's icky. It's not a lot of people want to deal with it, especially if you're talking about self-collection. You do have to navigate a lot of safety issues, especially when it comes to effective storage. Another issue is diet. As well as stress. So one of the great things is that it can tell us a lot about stress when we're looking at cortisol in fecal matter, but it can also, depending on your research question, be impacted by that stress. Things like clean water, irregular bowel syndrome, and then diarrhea can also affect your sample size, as well as diet. All of these things can impact your results. Typically, you can store it in -20 degrees. Unfortunately, how long samples last sometimes is unclear depending on the way that it's stored and also on what biomarker you're looking at. Generally, though, a lot of these concerns are in animal models, especially when they're collected like out in the wild or out in the field. It is stable for less time than blood. So, if you're thinking that 10 years from now, you want to go back and look at a question with fecal matter, it might not be possible depending on what you're looking at. So we're not going to talk too long on sweat, but I do want to bring it up because it is a really awesome, fantastic emerging technology that uses the concept of molecularly imprinted polymers are MIPS combined with AI learning to basically be placed into a wearable device and track continuous release of, as of now, cortisol and testosterone. So there's definitely a lot of validation studies that are coming through with this. And it's also a very important factor whenever we're thinking of hair research. So one of the things that I tell people is, now that we know for sure that we can get cortisol and testosterone from sweat, asking questions, you know, when you're looking at hair about how much exercise a person gets, how much time they're out in the sun, how many times a week that they wash their hair, or how many days a week that they wash their hair, are really important. And one of the advantages to sweat is that it is a continuous monitoring of hormone through wearable devices, and you get immediate results. But again, it's very much so in its infancy. So if anyone wants to do research around sweat and how we're getting hormones out of sweat, definitely something to consider. So again, some of the cons is it's extremely novel. Getting your hands on that technology, good luck. The technology is still emerging, and it's not well validated, and it's proving very expensive to scale up. So, I mean, unless you're doing a study in which somebody is exercising heavily, it's going to be a little bit difficult to get all the sweat that you need. And as far as using like ELISA or mass spectrometry to get those results, not a lot of information on that, unfortunately. Okay, so this is a pros and cons list. You do not need to do a screen capture. I will send it to you. But this is one of the, you know, just a few of like a rundown of a few of the things to consider whenever you're looking at all of these different biospecimens, or all of these different ways to collect biospecimens, depending on if you're looking at DBS or TAZO, and what some of those cons and what some of those advantages are. So just a quick refresher. One of the great things about blood is that it is very well established and you can get a lot out of it. Ditto with DBS, well established, cost effective, and fairly simple. TAZO, like again, think of this as DBS's, you know, older sibling or more advanced sibling, whatever you want to call it. Again, very minimally invasive, and you do get a bit more of your biospecimen. Saliva, very, very, you know, if you are considering a study where you want an acute reaction, so you want to look at something that's happening over hours, you know, or minutes, saliva is probably the way to go, depending on the biomarker that you're looking at. And it can capture other biomarkers, such as, you know, like CRP, interleukins, all of that great stuff, but you're just not going to get, you know, as many biomarkers as you can get with blood. Hair is awesome. It's stable over long periods of time, easy to transport and collect. But again, keep in mind that with hair, you're not getting an acute response, and it is susceptible to events that are occurring over those month-long periods of time. You know, but if your question is basal, if you're looking for a trait characteristic, if you're looking at the results of an intervention and whether they continue over time, hair is a great thing to look at, and not just with cortisol, but you can also look at things such as, you know, estradiol, progesterone, let's see, testosterone. Most steroidal hormones, I think, at this point have been validated. Don't quote me on that. So if you're looking at developmental changes over time, it's a great thing to look at. Let's see, urine. Again, think of this almost, I mean, I wouldn't say exactly as, you know, not as blood, because you can get shorter time frames with blood, but with urine, think of it over a 24-hour period. There's some stuff that you might be able to get over hours, like oxytocin, depending on your experimental design, but over and all in general, think about 24-hour periods. Nails and feces, again, nails, compare that to hair. I would say that if you had to pick one over the other right now, go with hair, just because there's still a few things that we need to, you know, test when it comes to nails. But if you're in a situation where they don't have hair, you know, go with what you can get. And then, of course, we have feces. which reflects the gut microbiome and metabolites. So it is unpleasant, but there's a lot of stuff that you can get from it. I have a 5-minute break built into here. So we are going to take a break for about 5 minutes, which is great because we're on time with everything. During that 5 minutes, if you have any questions, go ahead and place them in the chat. And let's come back. I'm on Central Time. So if you are on CST, then come back at like 1:27. And if not, just set a timer for 5 minutes. I'm going to start again in 5 minutes. Okay. Okay, so there was a question in the chat. So if you have replied, I'm looking to use CRP and IL-6 to screen highly stressed participants. Ooh, okay. And to find some neuroinflammation with DWI. I cannot do blood. I found there are mixed opinions on CRP. What is your view? Do you mean mixed opinions on whether, like, how valid CRP is in saliva? So, I usually steer away from the controversy. Like anytime anyone asks me something about oxytocin in saliva, I always am like, I don't know. Look at the literature and have your own opinion there. But I think so. We've done CRP and we've done IL-6 in saliva. I can say I've only assayed it in the lab once, and it was a bit of a nightmare because we just kept getting a lot of invalid results. Oh, is my screen still shared? Can you all still see the screen with 5-minute break, by the way? Thumbs up, thumbs down. It looks like it's shared. Okay, great. But I think one of the issues that we had there was that the samples came from babies, and it came from babies where they had used the cotton swab. So there are validated kits, commercially available kits. I think Enzo and Salometrics might have them, that do look at CRP, and they do look at a ton of different inflammation markers. I would say if you can, make sure you're getting whole saliva to mess with to avoid some of that. But whenever we did it, and again, I think this was just because it was in babies and they used the cotton swab. I had to do a lot of post statistics to get things in range. And are you talking about using, like, are you thinking about using like ELISA kits? Because that's usually the most cost efficient thing that people use. Most people use ELISA.
Yes, we're gonna use ELISA. But for example, as you said, like, I'm trying to understand if it's best to use a salivate or if to use the actual spit in the tube method, just to make sure, because I've heard that sometimes the threshold is too low, so you actually don't find much. I'm doing adults, so it should be a bit easier. So you're also suggesting to not use the salivate swap.
Yeah, just if they can spit in a tube, just have them get that whole salivary sample. And that was the exact thing that happened with us, is we weren't sure if it was just because the babies were too young. We weren't sure if they just didn't have that much inflammation, or if we got interference from the cotton that they used to do it. So I would say, if at all possible, just get the passive saliva. And as I said, there are kits that are validated, they work just fine. As a tech that used to be in a lab, it was extremely sad by the results that we got from that. I say eliminate as much as you can and just get that passive drool, and make sure you're getting enough of it too. I haven't looked at the CRP I haven't looked at the CRP protocols in a while, but you can look it up. So look up like the salometrics protocol, because I don't want to tell you the wrong thing. Look up the salometric protocols, look up the ENSO protocols, and see how much saliva that they call for. And make sure you have enough for redos. So make sure you have enough, and if that first one doesn't come out and the lab needs to do it again, make sure you have enough saliva. Like generally with hormones, it's really small, like 15 to 50. So you don't need a lot, but I don't exactly remember how much for CRP. If you want to, just stay on afterwards and I can look it up, but you can also look it up yourself. Just type in CRP protocols, and so Salometrics, whoever has them, maybe TCAN might have them as well, and look at how much you need. But yeah, if you can, get that passive drool.
Thank you. I will look it up myself. Thank you for your help.
Oh, no problem. Okay. Right, we're going to move right along. Okay. Sorry, I'm getting my screen back together. Okay, so what we're going to do now is delve into the time factor. Speaking of time, what time is it? I'm going to run through this fairly quickly because I've been kind of hitting on it all throughout the talk. And I do want to leave a little bit more time for specific questions about design. So time, what are we looking at, and what hormones are best for what we're looking at? How do we capture diurnal awakening, considerations for diurnal awakening slope? So how many hormones how many biospecimens and samples do we need to collect the diurnal rhythm? How many do we need to really look at reactivity and recovery? So when the hormone goes up and when it comes down. How do menstrual cycles affect that? Affect our findings, and how do we control for that? As well as basal, and very briefly, we'll talk about ratios and coupling. Oh, your goodness, advance. Okay, so time. Diurnal. So what biospecimens do we typically use to capture diurnal rhythms? Blood and saliva. Those are the two that we typically go with. So there are special considerations. If your research question has a question about diurnal rhythms, you do have to consider keeping a daily diary. Why? Because your day can be more or less stressful than the day before. You might be, in more competition. Something might be going on in your life that's going to affect your testosterone or estradiol or what have you level. So one of the special considerations that you should be controlling for is having that baseline data and having daily diary data. And daily diary data is basically asking questions like, what did you do today? Was today more exciting than the other day? What's going on in your life that might change the results, of my experiment? Also having baseline data, and typically what we mean by this is you're taking diurnal data over a variety of days. So you're not just doing one day, you're doing, 2 to 4 days back to back. So you do have to get multiple captures. I used to hear my mentor refer to it as, you can do the Cadillac, where you get, you're making sure you're getting waking, you're making sure you're getting 30 minutes after waking, you're getting right before bed, and you're getting bedtime. But depending on your research question, maybe you don't need all of that. Maybe you just need to get that waking level. And when I say waking, I mean what the level is when they wake up, depending on your hormone. So these are definitely things that you need to think about whenever you're considering whether or not you even need diurnal. So statistic considerations, when we're talking about diurnal hormones, it's not straightforward. So it's not straightforward, and a lot of times you have multiple collections across many days. So what does that mean? It means that we have nested data. So typically you do want to use some type of hierarchical linear modeling whenever you're doing this. You can use area under the curve. You can use averages. But if you're using averages, do you really need the diurnal data, or do you just need one or two or three or four waking, waking levels? Again, you want to make sure that you are, you're not doing too much, depending on your research question. One of the biggest advantages, though, is that diurnal statistics are rich in data. You get the waking levels, you get the CAR. I'm not going to talk about CAR or waking response because that's just not my level of expertise. But there's a whole battery of literature on TAR and CAR and what that means. You also get a slope, and arguably you also get a trait and day-to-day state level of an individual. By trait, I mean, depending on what that person's doing that day, if that's a part of your design, you have a unique ability to get, within person changes. So rich in data, great way to go. But again, you want multiple time points, and you usually want these multiple time points to happen across a few days. So I put a lot of references in the notes for y'all to have. But the rule of thumb is that you want to, when looking at diurnal collection times, even though I've seen it with waking and at bedtime, you'll still get a slope. But what you typically want to get is upon waking up, before leaving the bed, 30 minutes after the initial sample, before breakfast and before brushing teeth. And why? Because we don't want breakfast to end up in those tubes. And more importantly, we don't want to get blood contamination from when a person brushes their teeth. The reason why this second the second collection is so important is even if you aren't interested in the awakening response, it is a good idea to test for and control for whether that awakening response is impacting your diurnal curve or your diurnal slope. I'm sorry. Then you want to get one at a minimum 1 hour after lunch and at bedtime before brushing the teeth, again, to avoid blood contamination. This, again, is assuming, before, we've seen it actually with, even a midday or like mid-morning or mid-evening sample. But this is typically what you want to get. I've heard it, again, referred to as the Cadillac of sampling. But reality is, if you're doing this over 3 days, that's a lot of samples, right? And it's also a lot of money. So before you decide on, whether or not diurnal is the way to go, if I had infinite money and I also knew that all of my participants were going to adhere to making sure that they were waiting for those 30 minutes, getting the time variable in there, this is hands down the way that I would collect the data. If I were working with a bunch of kids or teenagers, I might highly change, my research question, or whether or not I need to have that diurnal slope, or whether I can get that trait level that I'm looking for, depending on my question, just by getting that waking sample. So, when to use diurnal, it can be costly. So if I were an undergraduate and I had a question that specifically had to do with diurnal and I wasn't working in a lab where my professor already had diurnal data, I might change my experimental design depending on, what was available to me. And I've said that a lot of times, there's a lot of diurnal data out there. If you have a question where you already, where the data is already out there, go find that data and hopefully it'll be able to answer your question. But it's going to be costly because again, the way that it should be done is over multiple days, not just one day. So it's good whenever you're looking at day-to-day changes from like a stressful event. Maybe you're looking at, whether we can get that, a different waking response or, something of the sort on test day. Also whenever we're looking at chronic stress or states, one of the great things about diurnal rhythm is it is, correlated to a lot of different health outcomes. And generally what we see is the flatter that slope, the worse the health outcome is, and the steeper that slope, the better the health outcomes tend to be. So it's definitely, if you can get that diurnal data, get it. Also, when participants are likely to adhere to collection, this is such a big deal. Even if, the timing, there are papers, I did not put it in the notes, but I will add some to it before I send them out. There are papers that show that even if you're off by as little as 5 to 10 minutes from the time that the person said that they took their, saliva sample to the time that they didn't, it changes that slope enormously. So you want to make sure that you're working with a population that's going to adhere to collections and spend a lot of time with thinking about that. So MEMS caps, which are caps that tell you when the participant takes the cap off to get the tube to give you their sample. I've seen people do it with selfies. So teenagers, take a selfie of them, taking the saliva sample, and you have a timestamp there. What was it? I think Emma was saying in her last talk, though, that you get some interesting pictures with that. But either way, you want to make sure that you're paying attention to time. Rich in data, rich in information, but it can be costly. So when to use reactive? Is it stressful? Are you looking at a competitive thing? Are you looking before or after a sport? Are you looking at them jumping out of a plane? Are you looking at them, I don't know, going through a sensitivity training, what have you? That's when you typically want to look at reactivity. And you also have to make sure that the hormone that you're interested in is reactive or passive. So, if you're interested in DHEA or progesterone or something of the sort that might be more passive than reactive, that's not the way you generally want to go. And also you want to make sure that you can get basal data, level data. So, I have seen and have questioned myself, how do you know that this is, reactive towards this specific task, or that this just isn't the way the person is? There are ways out of that, but typically it's a good idea to be able to capture basal data as well. It can also be used for physical or emotional pain. Lots of studies that look at that. But the gist of this is you want to get reactive hormones whenever you are doing an experimental design where there is a specific event. So not something necessarily that's chronic or over time. You still want to get 4 samples. And when I do say basal level, I've seen this happen twice. Typically what you want to do is on a day, preferably not the weekend, have a participant take these same samples at the same time, that are around the same time, at least, that your experiment took place. So that's basically to say that on a normal day, we didn't see reactivity. On this day, we saw reactivity. So this is just, this is a TSST, so the Trier Social Stress Test in children, and the way that they mapped out whenever they took their hormone samples. And so typically what you want to do is do it immediately after consent. If you're looking at stress, if you're looking at testosterone, if you're looking at some of these hormones, if you're looking at even, like oxytocin, you typically want to, have them rinse their mouth out. Then you want to get that consent done, have that mouth rinsed out, and wait for approximately 10 or so-ish minutes after that mouth rinse. And the reason that we want to get a hormone sample immediately is because sometimes people have a reaction just from coming into the lab. So if you're doing a reactive study or if you're doing some type of protocol where you're, introducing some type of variable, you want to get that first one very quickly after they come in. So in this case, they were using electrodes and looking at ANS data as a part of the design. And so they got one approximately 30 minutes after the lab arrival. A lot of times what you'll kind of see is called like a lab effect or just an arrival effect just from participating in the experiment. Then they also got the next measurement 15 minutes after the start of the TSST, because depending on what hormone you're looking at, that's about how much time it's going to take before you can actually start seeing that reactivity from the task. And then, typically what you'll want to do is if there are surveys or questions that you want them to ask after you do whatever the task is or whatever the variable introduced is, have them complete some questionnaires. And then approximately about an hour after the start of whatever your task is, in this case, it was the TSST, you want to get that final one. So, and this is typically, where there might be a little bit of wiggle room with diurnal, maybe not getting that cortisol rate or weight gain response or what have you, or getting that second to last response. Typically with reactivity, you want to have this. I think we worked on we're working on a hot sauce study now where we might be doing it a little bit differently, not getting that immediately like after consent, but typically that's what you still want, that after consent. I think in the beginning protocol or the pilot, we just did 2. But for publication, you're gonna want to see 4. Okay. So the other reason that you want to make sure that you get the basal day is to control for menstrual cycles, or typically what you can do is just make sure that you're getting all of the women to come in at the same time during their menstrual cycle. Keep in mind too that also men can experience fluctuations in hormones, which is why we go back to that basal day and getting approximately that same time for each participant. You do want to track whether or not the individual that you're looking at is on birth control or not. And if you have, again, a question that's more of a trait and not state, then you might want to consider other variables such as waking levels in hair. Why? Because a lot of times, not so much, like with waking, again, there are some articles to look at as far as like waking and menstruation, but you can get rid of that variable if you're using like a broader time period. So using hair, for instance. So when we are looking at basal levels, again, we are looking at things such as hair, nails, teeth, waking hormone levels, and averages of diurnal. Because again, depending on the model that you're using, you might want to take an average to have that trait level hormone, because that can maybe change the way that this diurnal curve works. So if you're getting diurnal, you're also going to get, basal, and you're going to get that trait, that trait hormone data as well. So for the most part, if you're looking at ratios and coupling, and what that means, if you're looking at the relationship between stress and development, you can do that with both diurnal and state models. So you can use reactive models. And I mentioned this because I've spoken to people where they only seem to think that you can get this if you're looking at diurnal models, if you get that crosstalk between the HPA and HPG axes, and that's not true. You can definitely look at ratios and coupling models just by using trait data and reactive models as well. So other assay considerations is our costs. So we are going to, so cost, access, biomarkers, and your biospecimens. Look at time, so yeah, 10 minutes. So we're gonna quickly kind of go through these because they are important. So we don't need to all know in complete detail how these assay processes work, but it's good that you have a general gist. So you can use our RIAs, so radio immunoassays, and these determine the concentration of an antigen, so hormones, proteins, whatever you're looking at in a sample. And it basically involves antibodies and radio label isotopes. The unbound isotopes are removed from the well with washing or aspiration, and a known volume of the sample is added to allow for the unknown amount of antigens to band to the antibody. So as the number of bound antigens increases, the radioactivity in the well decreases. So succinctly, basically, it's exposing the amount of a molecule to by sticking it to its counterpart, and what doesn't stick is washed away, and what's left over is what we compare to a known value. So these are the list of pros and cons. The ones that I want to point out specifically are that they're highly sensitive and specific. So they're able to detect very low sample concentrations. So remember I was talking about nails. When using antibodies is of a high affinity. Also, it's able to look at, peptides, drugs, enzymes, viruses, bacteria, tumor antigens, and other organic substances of biological interest. So very, very broad on what you can look at. It does require gamma counter and exposure to minimum amounts of radioactive isotopes. Also, they degrade over time, so there is a shelf life whenever you're dealing with these, as opposed to, some samples where, you can still detect some of these hormones years later. Let's see. Then we have EIAs, or enzyme-linked immunoabsorbent assays, ELISAs, which hopefully a lot of us are familiar with. And is based on the same principle as IRAs, but instead of labeling your target biomarker with a radioactive isotope, it uses color changes in the substances that are labeled. And broadly, basically, the antigen and the antibodies are incubated, just kind of like developing film. It sticks to the antibody. We again wash everything away, and we use light to read how much of what we're interested in is left over. And you have competitive and non-competitive ways of doing that. I'm going to leave this slide in here because it gives you a lot of information on how it works, but it's not necessarily relevant to how you pick out, decide on your study design. But in general, it's a very easy process. You prepare your sample. Typically, there's not a lot of extraction involved if you're using a steroid hormone, but if you're using a larger hormone, such as oxytocin, you are going to have to go through an extraction process. Typically, if you're looking at like testosterone, cortisol, DHEA, 2 hours to 8 hours, depending on how long the incubation is to get results. So some of the pros, it's simple to learn. You can use this and send it off to, professional labs that do it. You can find a collaborator. There are quite a few labs out there that are able to do this. It is expensive to develop new methods. And one of the biggest things that you do have to be concerned about is that it's only after the run that you notice things, go wrong. And it can be a serious issue if the biospecimens are irreplaceable. So while some of these techniques might be really expensive, if there is no way in the world you're ever going to be able to get this data again, maybe ELISA isn't the way to go. We also have lateral flow immunoassays. which is a popular method for detecting a measurable amount of the target molecule. These are pretty awesome because we're all familiar with these. And when I say all of us have probably taken a COVID test. And if we haven't, we're all familiar at least with the way like a pregnancy test is. That's lateral flow technology. Simple, very user-friendly. However, it is still emerging. There are some companies out there where you can use it with cortisol, but as far as validation with other hormones, there's not a lot out there. So, if you're looking at like like growth hormones, ACGs, things of that sort, a lot of stuff is, out there. It's validated, it's commercialized. But if you're looking at testosterone, good luck. It's not going to be something that you can use that I know of yet. Then we have liquid crow or LCMC or LCMSMS. And basically, it's a very specific measurement of hormones due to the direct measurement of mass. This is really, really awesome. I love this technology. And the main principle is that the sample is basically put through a mobile phase, and then an ionization process takes place. And then as a known precursor, it travels through it weighs it, and there's a relative abundance of precursors to producing the ion that's quantified. So it's very specific. It detects things on, based off of the molecular weight. So it's really awesome when samples are limited. Development of new methods is relatively simple, and can be completed rather quickly. And also you can test these things really easily before. So, it's not like you're doing it, there might be a problem with the curve, and then you don't know if your result's going to come out the way you want it. But it's complex. It takes specialized training, and it requires a lot of maintenance. So these are not inexpensive at all. So I have a list here of considerations whenever you're thinking about how, once you get your biospecimen, where you're going to, get the specific biomarker, some of the pros and cons. I always say, shop around, see how much it costs to get, these done in different labs, and see what works best for you. But also keep in mind, again, the amount of sample that you're actually able to get, and whether or not you're able to get that sample again. So how do you decide? What are some questions that you need to consider? You have about 2 minutes left. So what is your question? Is it longitudinal? Does it take place in a lab? Are you looking for chronic variables? How many biomarkers? What is your budget? How feasible is this to do? How many collection points are you going to have? If it's reactive, you're going to have those 4 points. Are you collaborating with someone, or is it all out of pocket? So while you might have this grand, wonderful design, can you get that data from somewhere else? Or are you in a situation where you might just be able to look at waking hormones? What's your population? So you might have a lot less restrictions when it comes to animal models as opposed to humans. What's their age? Whenever you're thinking of children and babies, be very, very, very cautious. And how you're going to be able to collect that sample. Some babies come out and they don't have hair. So are you really going to use a hair design where you're looking at, the amount of exposure to hormones, in utero, if that baby doesn't have any hair? And can you safely store the samples? And basically, are you able to also budget in transporting those samples? Okay. And so we're pretty much going to finish up, but at the end of your slides, I kind of just wanted to put a nice, again, this is at the beginning, but collection costs, storage costs, ease of collection and transportation, a rundown of the different methods that you should consider when things are being assayed. as well as whether or not you can get acute reactivity, diurnal or basal, as well as how complex that statistical analysis is. You know, sometimes cleaning that, if you're on a time limit, like thinking dissertation or what have you, it's a big deal to consider how long it's going to take you to clean and analyze that data. It is a lot easier to clean one sample than it is to clean and organize 24. Finally, when in doubt, make sure that you're reading the literature before you begin. Feel free to send me an email. I'll at least put you, like, you know, send you in the right direction. And make sure that you're collecting your control variables. So for each one of those hormones, and you know, we could talk about this all day, there are gonna be things that you wanna control for. Whether that's a daily diary and daily mood, or whether that's how much a person washes their hair. Also think it through, then think it through again, and then think it through again, and then talk to a bunch of other people on whether or not you've thought it through. This is really important. You know, once you do that IRB, once you've collected the data, it can be sometimes really difficult to go back and get that data or that control variable that you didn't consider. So make sure you're thinking through your design before deciding on your biomarker, as well as your biospecimen. And I am a minute over time, so if there are any questions, feel free to stay on and ask them. I will stay on for a bit longer. I think we can, Elizabeth, are you still there?